So what is gene therapy? Gene editing is one of the most controversial fields of modern science. Some forms of this revolutionary technology are widely accepted, like when it comes to the manufacturing of insulin, but other uses such as GMOs and gene therapy are generally shied away from the public. We here at Peering Into the Gene Pool aim to shine some light on these subjects. Check out GMO foods and organic foods at www.peeringintothegenepool.com. Gene therapy is the modification of an individual's genetic makeup by inserting new genetic information into cells to either cure diseases or repair defective genes. While gene editing refers to any DNA structural change, gene therapy refers to the use of genetic editing to produce therapeutic gain. You might be wondering how this is possible, and until recently, this technology was insufficient and out of reach for most scientists. In 2012, Jennifer Doudna and Emmanuel Charpentier published findings for their research on CRISPR-Cas9, a technique that would become the gold standard in genetic editing and would earn them a Nobel Chemistry Prize in 2020. The CRISPR-Cas9 gene editing tool was developed from microbial immune systems that protect the organism against viruses. The system is capable of locating specific genetic sequences and chopping them up. In bacteria, this means that rogue genetic material originating from viruses can be located and destroyed. Scientists can also use this targeting system in humans to locate specific gene sequences to cut it out and replace with the desired DNA sequence. However, there is a difference between somatic and germline editing. Somatic cells refers to all the cells in your body except for sperm cells and egg cells. Germ cells, or germline cells, on the other hand, refer to sperm and egg cells. The distinction between these cells is important when it comes to gene editing. Any changes in your germ cells will be carried on to the next generation and be permanently etched into the genomes of our descendants, while changes in the somatic cells cannot carry over in sexual reproduction. Another important consideration is that when a zygote, the combination of a sperm cell and an egg cell, is formed, that single zygote will undergo mitosis and keep splitting into daughter cells until a fully grown fetus has formed. This means that any alterations in either the sperm cell or egg cell that form the zygote will be present in the following cells, which is concerning when the long-term effects are still unknown. It is important to know that this is not the same as the new COVID-19 mRNA vaccine. This does not involve gene editing at all. For more information on this, feel free to read our conspiracy theories article on the anti-vax movements at www.peeringintothegenepool.com. Gene therapy involving somatic cells carries less risk than germline editing. Apart from the biological risk, there are also important philosophical debates concerning germline editing. We can decide for ourselves if we accept the changes made through somatic gene editing. But in doing germline editing, we are deciding the fate of those who are not yet born. These philosophical debates are important to regulate pitfalls such as gene doping and designer babies. Now let's consider for a second some of the pitfalls. Consider for a moment the possibility that we might one day have perfect control over our genes and those of our children. What if the technology becomes commercialized, that you would be able to simply order what you want in terms of genetic attributes? The best athletes would not have been those who trained the hardest, but rather those who had the money to afford the better genes. Lines would blur between what makes us unique, while the gap between rich and poor will simply increase. Could society collapse into dystopia where money is the one and only ruler, where the rich will become the smartest, strongest, and fastest, while others are left behind? Luckily, this level of control is not yet possible, but it is important that we remain aware of the dangerous possibilities. Instead, scientists aim to use this technology to further the field of gene therapy to eventually eliminate debilitating genetic diseases and improve medical treatment for patients in need. This could mean that people become less susceptible to common diseases or even immune to genetic diseases. These are exciting times for modern medicine, and we here at Peering Into the Gene Pool are looking forward to discussing gene therapy in further detail with you during the months of September and October. Join us to learn more about CRISPR, as well as some gene therapy experiments that have been conducted using this technique. We will be exploring both the positive impact that gene therapy can have on society, as well as the pitfalls that we need to be wary of while further developing this technology.